a big welcome to what we've called the past, present and future, and the lecture by Dr. Richard Paisey, who many of you do know. So if we go on to the next slide, Kerry. Okay, so big welcome to everybody. I won't do it in foreign languages for obvious reasons. So introductions, hopefully most of you will know Dr. Richard Paisey. Many of you will know Lexi Line, who is an ex-trustee of ASUK and will be presenting a little bit later. Johnny, some of you may have met, and he's, as you know, or as you may not know, he's doing some research around the eye at Great Ormond Street and other places. So next slide, please. Thank you. So very warm welcome. It's our 25th anniversary. So ASUK has been going 25 years, and hence trying to look at the past and what we've learned from that. And Richard's going to do that a little more. And today marks World Alstrom Syndrome Day, and Carl Henry Alstrom, born on the 3rd of May, 1907. So explore a potted history with us. Let's think about the past and what's worked. Let's look at the present, and let's look towards the future in the next hour and a half-ish. Okay, so next slide, please, Kerry. So I'm going to pass straight over to Dr. Richard Paisey, and he's going to both introduce himself and introduce this lecture on this kind of very special day. Thank you, Richard.
Okay, well, thank you very much for inviting me to speak. And I'm going to talk about the historical perspective of Alstrom syndrome. It's been a very great privilege to be involved with this. And so two aspects. First of all, the historical perspective I'm going to set in the context of genetics and our development of understanding of genetics, and also why collaboration's vital. So next slide, please. So here's the genetics timeline. We're straight in to this, and the thoughts about genetics and heritability of disease go back a long way, and around about 200 AD. Jewish and Arabic texts separately addressed the issue, well, boys with bleeding, that funny thing, which was haemophilia, seems to run in families. And there are many, many other examples of people and societies and cultures pondering on that thing. But it wasn't until 1865 when Gregor Mendel, an Austrian monk, really by experimenting with peas, height and color of peas and how they were inherited when he cross-pollinated pea plants, developed the laws of inheritance. Over that same decade, Darwin and Wallace were separately discovering the survival of the fittest and developing the theory of evolution, the half a billion years of development of life on the planet, effectively. It was a long time then, 1953, before Watson and Crick in Cambridge and Rosalind Franklin in London started to piece together, well, what was this substance in the nucleus of cells which passed on the genetic characteristics, the heritable characteristics. And it's called DNA because they knew the chemical structure, but they didn't know how the whole thing fitted together. And that was their work to discover that. Then at that time and a bit later, Fred Sanger in Cambridge found both what was the actual chemical building block structure of a protein, it was insulin, and the DNA that coded for it in this magical substance in the nucleus of cells that passed on heritable characteristics. So then the thought arose, well, okay, there's this code, this chemically written code in the nucleus of all cells passed on from one to the other and from generation to generation. How does that code make the cell make all the proteins that make up the organism or the person? There must be some messenger. And Jacob and Monod, French researchers, discovered messenger RNA, a molecule that flowed from the nucleus into the cytoplasm or the cell sap, so that the cell could then make the proteins it was directed to do. I've put a few more highlights here. 1978, this group of people, Winter was the British person who was involved in this in Cambridge, found monoclonal antibodies. So they're able to use the DNA understanding to make mice make antibodies to particular molecules, particular proteins. And that led the way for the monoclonal antibody therapy. So if anyone knows of therapies that end in MAB, you're having a monoclonal antibody therapy. And that enabled very precise targeting of cancers, of inflammatory diseases like rheumatoid arthritis and some liver diseases. Kary Mullis then was the next up, and he began to think, well, DNA, the code for inheritance, is copied as cells divide. So each daughter cell has the same copy of that DNA. How does that happen? There must be something that does that. And so the polymerase chain reaction enzyme, which duplicates DNA, or PCR, was discovered by him and harnessed by him and his associates. And you've probably heard of that in crime scene investigations, in inheritance studies, who am I, where do I come from, and so forth. So it's ubiquitous, the use of that, really. Next along, Barker and Hales, and Barker's from Southampton, Hales from Cambridge, and they sought to discover what genes you have, or could the genes be altered by environment? So they switched on different proteins according to the environment, and that they proved. 1996, Ian Wilmot in Edinburgh, he cloned Dolly, the sheep, and then in 2012, Charpentier and Doudna, French investigators, discovered a really important thing, which is so important for the future of genetic investigation and treatment, which is called CRISPR. And what they began to think was, well, if a virus gets into a cell and makes that cell make its own protein, it must be getting into the DNA. How does it cut into the DNA of that person or animal or individual or bacterium cell? And they showed how it did that, but even more importantly, showed how to harness that. And that's called the CRISPR technique. And next slide. Thank you, Kerry. And so this is, who are the discoverers and innovators who've done all this? A little bit more colour and background to them, underlining the need to collaborate. Gregor Mendel was an incredibly brilliant monk, student, scientist and monk, and his abbot was absolutely keen in the 1860s to further the study of, in monasteries, particularly his monastery, of scientific investigation. And Mendel set about with his gardener, studying thousands of experiments with peas and beans to see whether the height and color of the next generation of beans could be determined by the way that he cross-pollinated them. So in so doing, he basically discovered the laws of genetics. Over that same time period, Wallace and Darwin both discovered or got the theory going, really, of survival of the fittest, and then building on that, the origin of the species and evolution. And they were very different characters, but co-presented and worked together. And Wallace was a religious person, and Darwin had a terribly sad event in that his favorite daughter, Annie, died aged 10 of pneumonia, and that colored his whole feelings about what it meant the survival of the fittest in evolution, whereas Wallace thought, well, what it means is we've got to try and help everybody. Everybody's the same, all races, all people, they're equally important. And so that debate, if you like, between science, religion and society is an abiding one. And it's present with us today, of course. Then Watson and Crick, here they are with their double helix, and Rosemary Franklin was the one who did crystal studies, which helped them. Jacob and Monod with Sidney Brenner. So here's a collaboration of rivals, and they just couldn't find this messenger RNA. It was such a fragile molecule. It kept disappearing in their test tubes and their experiments. And after a long conference, they spent all day Friday, it was Good Friday, together, as it were behind closed doors, like jazz players doing a jam session just for themselves, until finally Sidney Brenner stood up and shouted out, "It's the magnesium," because he realized its key ingredient was missing that would stabilize the molecule. So that went from there. Here are the monoclonal antibody guys, Colliner and Milstein, and Winter was the British man who worked with pharmaceutical companies to make them into drugs used for people. Here's Fred Sanger, the one who got the structure of insulin, the structure of the DNA for insulin. He's another very humble man from a Quaker background. And when he retired at 65, they said, "Well, are you going to head up some big science institute now, or get lots and lots of money from lecturing?" He said, "No, I don't think so. I've been pottering around in a lab all my life. I think I'm going to go now and do my garden." And that's what he did. Here then are Barker and Hales, and their muse was called Ethel Margaret Burnside, and she was a formidable lady who had a set of data on which their work was based. Carrie Mullis discovered the PCR, and Ian Wilmot, here he is with Dolly the sheep, and here are the two girls, Charpentier and Doudna, who with many big pharma collaborators got the CRISPR technique, editing of DNA, possible. Next slide, please. Right, well, here then some key images. This is the pattern of autosomal recessive inheritance, so that if two parents each have a mutation in a recessive gene, the other copy will work, so they won't express the disorder. And that's the same with this next generation. There's some heterozygotes, that's to say they've got one copy of the mutated gene only, whereas the next generation, two of the four siblings have inherited both sets of mutations, so they express the disorder, and that's recessive condition, typically Alstrom. Thomas Hunt here, just mentioned because he actually visualized chromosomes and counted them for the first time, and he used these little creatures called Drosophila, and they're fruit flies. So when you put all your peelings of fruit and vegetables in the caddy ready to go out to be recycled, and you open the lid and all these little flies fly out, there they are, they're the fruit flies, and they lent themselves very well to those studies. Now over here we've got Rosalind Franklin looking down her microscope, and there's the famous photograph 51, which is a photograph of her crystal of DNA, which with X-ray diffraction to show the protein and that it must be a double helix, and that's what led Watson and Crick to the discovery. Very sadly, Rosalind died in her 30s of cancer, so she didn't receive the Nobel Prize because it's not awarded posthumously. Here then's the DNA to RNA to protein, reading of the DNA to the RNA to the protein, and here's the amino acid sequence of insulin, which Sanger sorted out. And if you think about it, what that's meant is that the world's supply of insulin is assured because you can insert the DNA that codes for this protein into bacteria, and then those bacteria will make insulin, and you can harvest that from that fermentation. And so that's assured the world's supply of insulin. Here's Ethel Margaret Burnside, this really incredible woman. She's called a big bony woman, 5'10" tall, long thin face, dark hair, which she wore scraped back, very imposing presence and penetrating voice, a dominant personality. So she said to her midwives throughout Hertfordshire, of whom she was the head, what's this poor effort of just giving a woman a baby? They've just been through pregnancy and birth and given birth to this baby, and here you are as a baby girl weighing £7 6 ounces. That's it. Go away and breastfeed. It's we should be weighing the placenta, recording the time of delivery, if there's premature birth, if there's delayed birth, all these things, so that we can record this. And so she did, and she recorded this in the 1940s and 50s, and it was stored. And the managers of the facility, the Hertfordshire Maternity Hospital, saw fit to preserve that data. Next slide. So that was how. Sorry, could you go back one? Sorry. So that was how David Barker and Nick Hales were able to go back to those records that were preserved with the early NHS numbers, and then follow those persons when they were 50 years old to see if there'd been an effect on their genetic expression of their enzymes and their metabolism from particularly starvation in utero and being small for dates. And so there was. The ones affected by that had more diabetes, more heart disease, and more high blood pressure. So that was an extraordinary collaboration through time. And next then, so now armed with that, we're going to go on to the timelines in Alstrom syndrome. So these are columns going across from left to right. And first, 1958, Carl Henry Alstrom described the syndrome with many of the features of it. In the 1990s, the late Jan Marshall was already working on the condition, and she found the Acadian French population with about 30 cases. And I'll come on to that a bit more later. Then in 1998, John K. Parkinson founded Alstrom UK, the Torbay Clinic. There were 7 UK families at the time, and we used a hotel in Berry Head, a clinic in my one office, and people came from Portugal, Germany, Italy, and Georgia, in Russia as well, to that clinic for families. In the 2000s, Tim Barrett joined to look after the children. More changes in the condition which might happen to some patients were discovered. And then finally in 2004, the actual ALMS gene was discovered, the gene which, when mutated and you inherited both copies, gave you the syndrome. That was an incredible moment. And that was also collaboration, because it's discovered by Jan studying 30 or 40 cases and painstakingly working along the chromosome to see which ones were mutated, whereas David Wilson in Southampton, geneticist, had 1 case. And it was fortuitous that the ALMS gene that was mutated in this young person with Alstrom syndrome was broken. It had a translocation, it's called, a break in the chromosome. It was right through the middle of the ALMS gene, so he was able to identify it quite quickly with 1 case. But interestingly, they both submitted to Nature, and both of their submissions were refused. And the reason was that each had only half of the answer. They'd each only really properly mapped out half the gene. So the editors of Nature wrote back to them and said, you work together and collaborate, and then send back in your articles, and we'll publish both of your work side by side. And then from 2008 to 2013, we had what's called the DAS study, defining the phenotype in Alstrom syndrome, showing what the characteristics of patients with the syndrome are. And this is a lottery science grant. We had 70 families by then in the UK, and we showed a number of things by studies, which I'll come to on the next slide, please. So here we are with some pictures of the main protagonists. Here is Carl Henry Alstrom, a very compassionate man for the time of the 1950s, when doctors, I think, were known for being rather abrupt. He studied TB and epilepsy and their effects on the psyche, and was a very good psychologist, psychiatrist, and general physician, and above all, listened to patients. So when he saw these three cousins with deafness and blindness, obesity, and insulin resistance, he thought about it, talked with them about it, talked about their family histories, and worked out that it was a syndrome, and quite appropriately, it was named after him. Then Jan here, the lovely Jan, who we all missed so much, who did so much and embraced the patient's cause and them and the families. And she worked with Gail Collins, who was a person who came from the Acadian French families where there were so many cases. And the story is that in British colonialism, the French were displaced from Newfoundland and went to live in Louisiana as the Cajun people and back in France, but many were never happy there. And so they migrated back, and they had a lot of first cousin marriages. And so Jan traced back the founder effect there. Kay, John and Matthew and Charlotte Parkinson were fantastic people, without whom we wouldn't be a quarter of the way we are with Alstrom Syndrome UK, and with many studies and with much of what AS UK does. Here are the inheritors of this mantle, the Leeson Beavers wonderful family, and all that they've done that many of you will know. I've put here Kathy Carey, who did the initial heart studies, Tim Barrett, the paediatric lead, Pietro from Italy, who's collaborated so well with Jan and ourselves, Prof. Rob Semple, the stalwart, brilliant scientist, who was then in Cambridge, now in Edinburgh, a molecular biologist, and his collaboration was with a brilliant laboratory scientist, Zhang Hu Chen, who's obviously on the paper that was published from their cellular work, and it shows you a little glimpse into something that's so important, that any great scientist or leader or group, their work depends on the scrupulous, dedicated work of dozens and dozens and dozens of laboratory scientists, people who clean the labs, those who look after the test tubes and the temperatures of incubators and all those things. And without that collaboration, none of their work would flourish. Here's Rick, who we all know from his really cogent, down-to-earth talks, and the cardiology lead, and Tarek, who's led the clinic in Birmingham, and Ashish, I haven't got a picture, I'm sorry, who does the cardiology for children in Birmingham. And next slide. Now, so here again is how this developed in practical way. In 1998, Kay Parkinson came to the end of my diabetic clinic in Torbay Hospital and said, I've two teenagers, I want you to look after them. They're both blind and both have very young onset type 2 diabetes. And I said, yes. So we went from there, and it was clear quickly that Alstrom syndrome was the likely diagnosis. So rare, only a handful of cases known worldwide. And so without more ado, Kay's said, well, can we start a family support charity? Can these charitable funds be used to support families to be seen in one site in Torbay, where you're looking after Matthew and Charlotte? And we chose a hotel in Brixham. Can we integrate family visits into Torbay Hospital medical facilities? So we did. And is there NHS funding to support specialist clinics? And that was sought out again with the drive of Kay Parkinson. The next slide. So from 2005, specialist funding was obtained. So now it's top 15% of NHS funds, which is billions, is creamed off to directly support specialized services. So it might be heart transplant, live-related renal transplant, pancreas transplant, certain cancer treatments, and rare diseases like Alstrom. And that funding was obtained and exists still and has been well fulfilled in the Birmingham clinics. We established links with the Jackson Lab and Jan Marshall. Edmund Jessup was the lead of all these specialist commissioning services, and he came and reviewed, audit every year is done on these services, and he said to us, well, I'm really pleased. I want to encourage you to carry on with the research. We can't pay for the research, but it's an absolute integral part of what you're doing in doing the clinical support for patients. And if you can get funds and do research studies, that's exactly what we would like to see. Tim joined us to help with all this, and we had patient feedback after every 6-monthly clinic. And what I think is most important, I still work with specialised services in other fields, is that we, after the patient feedback, we got the letters to the patients and carers done in one day. And it's absolutely crazy to have a group of very clever professional people sitting around and meeting patients and families and coming to conclusions about them and then not telling them what the score is. That's absolutely awful to me. And I think that was one of the good things with Sue Wise and then Cheryl, my secretaries. And working with local teams was crucial. Many, Sheffield, Leeds, Winchester, Birmingham, Nottingham, those teams where patients lived could do a lot of the work with patients and liaise with us, and that made for really good sense and good outcomes. The next slide, please. So as this progressed, we thought about where did Alstrom sit amongst other conditions, and that's quite important, because if somebody's in a paediatric clinic and seeing that there's a youngster in front of them with nystagmus and blindness or impending blindness, that they might think from the family and things that this might be a recessive condition. So that would sit with cystic fibrosis, phenylketonuria, Bardet-Biedl, and Tay-Sachs, and many others. So these recessive conditions fit together, and there's Alstrom amongst them. Alstrom is a ciliopathy. The Alstrom protein that the gene codes for is part of 100 or more proteins which form the primary cilium, which is an evolutionary preserved cilium, like a hair on every cell, and it senses the outside environment, conducts the signals to the inside of the cell, and conducts signals through the cell. So it's a ciliopathy, and other proteins which, when mutated or not present, are cause disorders are Bardet-Biedl, Juber, and Usher syndrome, which is a deaf-blind syndrome only, primary ciliary dysplasia, which causes lung failure due to pulmonary fibrosis, Alstrom, and polycystic kidney disease, you may know, which is one of the commonest inherited disorders of kidney disease. It is also a ciliopathy. And then the other group, which may lead a clinician to think, well, where does this patient sit? And might look up the list and find Alstrom, is deaf-blind syndromes, of which there are 40, mostly inherited, but congenital rubella here clearly isn't. So there's Leber's and Bardet-Biedl and Usher syndrome. So those are the sorts of families of disorders within which Alstrom sits. And next slide, please. Now, how does a change in that gene, the ALMS gene, which then can't code properly for the ALMS protein linked with the cilium, how does it cause all the problems? What are the problems in tissues that are affected? And this next slide's a panel of histology slides from kidney, pancreas, liver, testis and ovary, lung, and the heart, and a section of the retina in a person who's affected by Alstrom syndrome. And the characteristic thing of all of these tissues, bar the retina, is that they're all affected by fibrosis. There's much too much fibrous tissue, scarring tissue in all of those organs. And we know from the way patients develop in their lives with Alstrom syndrome that not every Alstrom patient gets problems with every organ or all the things that could possibly happen, but there's a common thread here. So how might it be then that the lack of the ALMS protein causes these things? The retina is different, as is the ear, because in that case, the ALMS protein is part of the cilium, and that cilium in the retinal cells is crucially important for conducting and flowing proteins in the retina up and down from one part of the retina to the other. And if that doesn't happen, the system gets clogged up and the retina degenerates. So it's a direct effect of the lack of the ALMS protein. And next slide, please. So here we are then, we've got this is a flow diagram, Alstrom gene mutations. They cause directly eye and ear problems by the absence of the protein. They cause insulin resistance because insulin doesn't flow to the surface of the cell on all these tubules in the cell when glucose stimulates it to be released. So that causes insulin resistance, which leads to diabetes. This may cause fibrosis of other organs. So what effects would preventing that insulin resistance have on some of the other changes in Alstrom syndrome? And I'm going to touch on that in the next couple of slides. The next slide, please. Thank you. So here we are, and this is the first slide on, this is a panel, a set of panels of different outcomes from the DAS study, which 62 persons with Alstrom kindly consented to take part in. So this first panel is a young man who has diabetes, has high blood fats, high blood pressure, disturbed liver with fibrosis of the liver, and over a year with very intense exercise, walking, and diet, avoiding sugars and too much fat and eating a healthy diet altogether, lost 6 kilos. His diabetes was in remission. The disturbed blood fats went to normal, blood pressure went to normal, and the liver test of fibrosis reduced, and his ultrasound of the liver, which had previously shown fatty liver, went to normal. So we can say that tackling insulin resistance can achieve all those benefits, probably not the fibrosis in the heart, the testis, the ovary, the lung, and so on. Then the next panel is successful surgery. There were some tragedies with persons dying after surgery who had Alstrom syndrome from respiratory problems. So we were able to show that careful post-operative care, ovarian cysts, stomach problems, coronary artery disease, renal pancreas transplant, large bowel obstruction, spinal decompression and scoliosis treatment, hip replacement, and bladder dyskinesia, all pretty major operations could be done safely in Alstrom persons. Despite diabetes from adolescence, we noticed a striking finding that the feet stayed normal. People with Alstrom syndrome did not get numb feet, and did not get foot problems. Now, with the increase in obesity worldwide and the increasing numbers of young people getting ordinary type 2 diabetes at a young age, this is a very big problem. And in our cohort of non-Alstrom patients with young diabetes, 35% had numb feet, several already had ulcers, and they'd all had diabetes from 12 or younger. And so this is a stark difference, a very, very distinct difference. And that means something, and that's still yet to be followed up by a genius in neurology or molecular biology to find out quite why that is and what that might mean to try and prevent neuropathy in general. The high sugar, high carbohydrate diets were found to be worse for blood fats in people with Alstrom. And we also showed in this study that these incretin analogues like semaglutide liraglutide and so on that many of you maybe have heard of and be treated with were effective in Alstrom. We had an outcome measures of what happened to patients, and particularly found that really the death in infancy from cardiomyopathy could be averted. Death could be averted, and patients could recover from that serious cardiomyopathy, which is unique in cardiomyopathy in childhood. There were 2 anesthetic deaths, as I alluded to, and because pneumonia and ischemic heart disease were prevalent, there were 5 deaths from pneumonia in the cohort, and so we've recommended vaccination against pneumococcus and against flu from a young age. And we also showed in the DAS study that the cells that you kindly donated, which could be then cultured and developed into pluripotent stem cells, that they could be studied. And next slide, please. So here is that research, the promise of stem cell research, so that cultured cells could be, and this was done with the Alstrom cells, developed into pluripotent stem cells. That means cells, as in an embryo, which can develop in a whole variety of tissues. So what was done was to bone marrow, nerve cells, heart muscle, and pancreatic islet cells. And this is the work of Rob Semple and Dr. Chen, and will be pursued with a new mouse model. But if you just dwell on that for a moment, supposing you were able to culture large numbers of heart muscle cells from an Alstrom individual who had cardiomyopathy, you, with CRISPR technology, might be able to correct that genetic problem in those heart cells and then generate corrected cells which could be transfused or infused back to repair the heart. So that's not going to happen tomorrow, but that's one of many hopes for future. Next slide, please. And so here's great thanks to all of the people, all of you. Here's Trevor, the marvelous Trevor, who's been finance officer, treasurer for many years and is still collaborating with the board until he can come back and be a trustee again. And here are many of the families. And again, many thanks to all of you. And next slide. And here we are, 2 of my favorite people in the whole world, Lexi, who's going to speak to you next, and Kevin Goodwin, on the cycle ride. And this was Kevin's idea. And he said, well, if we're moving the clinic from Torbay to Birmingham, we should cycle it. And so here we are on the tandems, and Kevin pumped along at a great rate, so much so that his really quite high blood sugars went low, and we had to revive him when he had a hypoglycemic attack halfway along one of the days cycling. And that proved, as did Lexi, that exercise can, in the syndromic diabetes of Alstrom syndrome, be improved by exercise, just like anybody else with diabetes. So that's a big thing. And there's Lexi, and her now husband sitting on the front, said to me when we'd hurtled down into Exeter down a big hill, he said to me afterwards, Dr. Paisey, I want you to know we hit 45 miles an hour on that hill. And I thought, oh no, I was glad I didn't know that in advance. But we survived after many mishaps, going through a flood and getting lost quite a lot of times. So that was extraordinary and fantastic. Next slide, please. So this is a big thank you to everybody who's taken part in all this development of the clinic and the understanding of Alstrom syndrome worldwide, with hope for the future. And finally, I'm going to leave you with 2 references. I'll send all the references and the biopics, the text of the talks about each of the individuals we've we've listened to, we've heard, shared together. But this is current management of Alstrom syndrome and recent advances. Not so recent now, because it's a few years ago, but Kerry and I wrote this. And I think it's very important because we were able, with their permission, to harness and share the experiences of Alstrom families and the wonderful achievements of so many, many people living with Alstrom syndrome. And I think that's why it's such a good article. It's not on PubMed, so you can get it on Google Scholar and such like, but it's and I can send it to anyone who wanted it, a PDF copy of it. But I think that's very important, not just because me and Kerry wrote it, but because I think it's got that witness of so many lovely friends and patients. And this now is another one, The Secret of Life. So this is Rosalind Franklin's story, written by Howard Markle, and it investigates that controversy. Did Watson and Crick steal her work, or were they really collaborators? In the end, they were effectively collaborators, but it's a nice whodunit. And so with that, I'll say thank you so much for listening.
Perfect.
Bye-bye.
Perfect. Thank you so much, Richard. That was extraordinary and
Thank you.
Lovely way for us just to reflect on the extraordinary work that clinicians and families have done. You know, and if you just think it's only kind of 25 years ago, which is, you know, is nothing really. And the beginning of certainly AS UK must have kind of felt like walking up Everest in flip-flops. So thank you so much for that, Richard. It was extraordinary.
Very welcome. Thank you for inviting me.
Thank you. I am going to invite Robin Marshall, if he's still with us, probably just to say a couple of words, because there's quite a lot of mention there of Jan Marshall.
Oh my gosh, first of all, it's just fantastic to see everyone, and it's for me, it just evokes so many memories. When Richard was tickling feet in Georgia, I was a neutral test subject because I too have type 2 diabetes, and he got all sorts of good reactions from my feet. So it was pretty obvious that the kids weren't showing much peripheral neuropathy. So anyway, funny thoughts like that you might never think of again if you didn't have this kind of opportunity. And I would just applaud Richard's concept of the collaboration that's taken place over the years, including in the very early years when Matthew and Charlotte were part of one of Jan's early cohorts that also proved to Jan that it was not an Acadian disease because they were so obviously as British as could be. So things like that. And again, I have to applaud the efforts of Alstrom UK with enterprises such as this. It's a good example to all of us who care and all the organizations with whom we work. Bravo.
Thank you. Thank you, Richard. And hopefully many of us will be able to get to meet at the ASI conference in America in October. So,
Indeed.
Robin.